So we started with an introduction to animals, and we are going to continue that today, and then we are going to start with the lecture on invertebrate animals. So everything that we are going to listen in this part of the lecture, this chapter, is generalities that doesn't necessarily apply to every single animal group that we are going to review. For instance, most animals are going to have a larval stage, meaning an immature stage, but not all of them. For example, we don't have a larva, right? So those stages are for many other animals, but because we vertebrates are rather not that numerous compared to many other groups, especially invertebrates, that's why we say most animals are going to have a larval stage. Most animals also are going to have some type of metamorphosis, but we will see that metamorphosis is a term that only applies to a certain types of insects. We will discuss that in the next chapter. Now, something important is that all animals share this family of developmental genes. So among the most important developmental genes are the Hox genes, H-O-X. Okay, those genes in different numbers, depending on the animal species, are going to be very important to coordinate all the development since it is an embryo until adulthood. Okay, from embryo or when the egg starts to develop, as we saw in the last class, in the short video, until it is adult, there is all this coordination of maturity. And sometimes many of the same genes are going to be expressed at different points in time during development, okay? But every gene not necessarily produces the same protein. Sometimes a single gene produces a variety of proteins, okay? And all these activities coordinated by the non-coding portions of the DNA that we were discussing in the previous lecture, that we used to call junk DNA, which is very important, the non-coding, because it's the coordinating framework for the development of the end product, whatever that animal is. And among those, the Hox genes are the ones that are the most influential, especially during early development, okay? In terms of animals, we have more or less described more than 1 million living animals, but the estimates are much larger. More than 10 million organisms are suggested to be missing in terms of the animal kingdom. Now, in terms of the chemicals that are very important for coordinating activities in our body, the earliest evidence that we have of those, which are the hormones or steroids, are from sponges from over 700 million years ago. Okay, so that's the earliest evidence of those type of chemicals doing some activity in the body of an organism in order to coordinate the physiology. The common ancestor of all the animals, it's about 700 million years old, based on the evidence that we have. And when was the Cambrian explosion? Anybody remembers, more or less, when did the Cambrian explosion happen? Was earlier than that date or later than that date? 500 million years ago, so it was later. But the ancestor of all animals is older than the Cambrian explosion, okay? So don't confuse when the last common ancestor of animals appear in the fossil record with the huge diversification that occur in general in all the animals and in most organisms on the planet, which is a different period. Now, anybody remembers how many ice ages we have evidence for in the history of the planet?
What was the question about?
how many ice ages the planet has undergone throughout its history? More or less, remember? Somebody say 4 over there. Who needs more? Less? 3 major ones, okay? So, the earliest one is about a billion years old. The other one was in between 800 or 700 all the way to the Ediacaran biota or the Precambrian, less than 700 million years ago, and that is the famous one known as snowball Earth, because basically the planet, if you would be at that time on planet Earth from space, it would be like a snowball, okay? And that one ended up about that time when the last common ancestor of animals appeared in the record, over 700 million years ago. No, it ended even later, right before the Ediacaran explosion of diversity. That is about 650 million years ago, okay? Now, origin of multicellular animals. Now, in terms of phylogeny, what you are seeing in here is what we consider the animals. This is the clay, and the sister clay to all other animals, or what we consider animals, is the sponges. Now, the sister clay to all animals, it's a group of organisms, multicolonial organisms known as choanoflagellates. Now notice these organisms that are usually eukaryotic single cells, or they form these colonies. Look the similarity of this single cell to the cells of a sponge. We will delve into the details of those things. But you can start to see the similarities and the origins, the ancestors, and why animals, sister clade is the choanoflagellate unicellular eukaryotes. Now, for multicellularity to occur and to be true multicellularity, you need communication among cells, right? So they need some form of attachment to transmit information, and that occur only in the line of animals, about 700 million years ago. Now, there is a family of genes that is present in all the animals, and it is as old as that date, and that is the molecular evidence that we have in order to know how closely related they are to each other. And that family of genes produces proteins known as caderins, and those proteins, when you compare them, you put them together in a phylogenetic clade. You have here the choanoflagellates, then different groups of animals, and you can see that there is variation in these genes, the caderin or the caderin proteins, but there is a domain known as CCD domain that is found in all animals, and there is an analog domain in the choanoflagellates that basically shares the same function, okay, which is basically communication. Okay, production of some proteins that are going to allow the communication between different cells in the organism. Questions? No?
Historically, we have taught, or scientists have thought, that the last common ancestor of choanoflagellates in animals was a single cell organism. And so one of the transitions that between that ancestor and animals had to have been the acquisition of multicellularity. And I still feel that's true if we're talking about true multicellularity, where what I described, where a cell cannot survive outside of the exactly. So now I'm beginning to wonder, though, whether that last common ancestor might have been capable of forming simple colonies.
Okay. I just put that clip because it's very important that you keep in your mind that jumping from unicellular eukaryotes to true multicellularity is not an easy task. It's very easy to say, but it's not an easy task. So scientists that specialize on these topics spend a lot of time thinking on the processes and the steps that occur through evolutionary time in order for true multicellularity to occur. True multicellularity means, for example, that any of your internal organs cannot survive on its own. It's part of a whole system, and that requires specific adaptations, especially in the communication system of the multicellular organism. Okay? Questions? Ten more seconds. Okay. All right. This period in the Proterozoic that goes from 1 billion to 441 million years ago is known as the Neoproterozoic as well. And within that period, by the end of that period, closer to the Cambrian, is when we find this famous Ediacaran biota. And it was the first spore of diversification of organisms, and is when we have the earliest fossils of multicellular animals, and most of the fossils that we have uncovered suggest that they were invertebrates. They were soft-bodied organisms, okay? Some of them resemble mollusks and some sponges, some nidarians. We will cover all those groups, so don't worry in trying to understand which they are, but mostly invertebrates. But the problem is that with any other fossil, if it is not a fossil that gives us a real body part, it's very difficult to figure out the structures just out of impressions of rocks. But in any case, some of these fossils have provided extra evidence that resemble some types of developmental stages in them, at least on the impressions that have been found. Some other fossils, what they suggest is that predation started early on. So this fossil over here is also from the Ediacaran era period, and what you can see in there is a hole that is very similar to some predators that we have living nowadays, that basically they perforate the shells using like a drilling tool in order to penetrate the defenses of the prey and then consume the prey. So there seems to be this type of record in fossils that suggests that predation started early on in the development of life. The Paleozoic era, which is where we have the famous Cambrian explosion, started about 440 million years ago, and it ended with the biggest mass extinction at 250 million years ago. Anybody remembers what is that biggest mass extinction that ended the Paleozoic era? The one that wiped out almost every living organism on the planet. 95% of all organisms disappeared, more
Permian?
or less. Anybody remembers? The Permian extinction. Okay. about 250 million years ago. That is the extinction that ended the Paleozoic era to give rise to the era of the dinosaurs, okay? The Mesozoic era starting with the period that is known as the Triassic, okay? Now, getting back into the Cambrian explosion, 430, 425 million years ago is where we see the largest diversification, and is that period where we see the development of certain carbonated skeletons, certain animals with hard parts, and also when most or all the phyla of current living day organisms were developed, okay? Other important thing of the Cambrian is that the animals that we know as bilaterians appeared during that time. Bilaterians mean bilateral symmetry, just like us, right? You divide the animal into parts and you get two equal parts, quote unquote. We will see that that is not completely accurate. Now, here is where we see also the development of complete digestive systems, because if you remember, earlier forms would just do the exchange and absorption of things that they needed to do via diffusion, okay? So what is being suggested for the rapid diversification during the Cambrian explosion? One has to do with ecological interactions, which makes sense, right? So competition pushes natural selection, evolution to really produce adaptations in order to survive. So having a lot of competitors and also predators is going to push evolution to exploit new niches where organisms can adapt and avoid to being eaten or to be outcompeted for resources. The other one is the rise of oxygen levels. If you remember, anybody remembers when we start to see a little bit of increases in atmospheric oxygen in the history of the planet? When did that increasing atmospheric oxygen start? It's very long ago. It's over 2 billion years ago. 2.4, 2.3 billion years ago, but it was tiny amounts. But it's when we start to see that increase. And then in this period is when we see the first rise of oxygen, that it is represented, I think, about 11% of oxygen in the atmosphere. So that opened up opportunities for the evolution of new organisms to exploit the new niches of oxygen. Remember, life arose in anoxic conditions. So oxygen became a new niche that needed a new set of tools for the organisms to survive. Now, the other idea is the evolution of these new functions of genes in the genomes, okay? Because we are talking about very simple genomes at the beginning of life on the planet, and then as life got complex, more and more additions into the genomes started to accrue. And many of the novelties of the genome didn't come so much as the production of new proteins, but in the regulation of the machinery that was already in place. Think it, if you're going to think it in a simpler way, think on a factory. There are factories that only produce shoes. There are other factories that can produce more than one thing: shoes, pants, shirts, whatever, right? So that is kind of a flexibility, going just from shoes into all types of clothing. Think of the same in terms of the genome. The genome was very simple, producing simple life, then it started to produce more complex life and more complex life, and for that you need a different set of instructions and probably part of the same, if not the same machinery, used in a different fashion, okay? So think that analogy, if that helps you, to see how improving the complexity of the genome and the regulation of the genome by different proteins, regulatory genes, non-coding portions, are going to be able to produce new adaptation and the evolution of new organisms. Okay? Questions? Okay? Few more seconds. Okay, so we said that everything, the increasing diversification started with the Cambrian explosion, but the Cambrian is just the first period of the Paleozoic era. And the Paleozoic era lasted for about 250 million years, okay? Many different periods in that part. Well, diversity continued to increase during the Paleozoic era. And animals started to make an impact on the planet or appear, let's call it in earnest, on the planet about 450 million years ago. That is not the Cambrian. That is the Silurian, Anthropordovician, the Silurian periods, and also part of the Devonian. And the first ones to really colonize land were the arthropods. About 300 million years ago, that is the Devonian, Carboniferous periods. And it was some adaptations that we will discuss in more detail when we reach the arthropods, but they have to do with the hard exoskeleton that was able to support them on land, okay? We have mentioned this. Let's move into the era of the dinosaurs. Mesozoic era, 250, 66 million years ago. And something important that we have not mentioned this far, because the most famous things in that era are the dinosaurs, and what group of plants was dominant during the Mesozoic era? That was right before the Mesozoic era, during the Carboniferous Permian, which is Paleozoic era. Seedless vascular plants, yeah, like monilophytes, yeah, that's true. But what started to appear in there at the end of the Paleozoic era in the Permian, Carboniferous Permian, that was still not dominant? What follows vascular seedless plants? Yes, gymnosperms, okay? So, and those were the dominant groups during the Mesozoic era, the era of the dinosaurs, okay? Other important group that started to develop a lot during the era of the dinosaurs were coral reefs. And as you know, they are the rainforests of the oceans, and they are very important for the production of new ecological niches that allows what? Diversification of new forms of life, okay? The reptiles, it is interesting because the dinosaurs, just like mammals, we come from aquatic ancestors. But during the Mesozoic era, many reptiles came back to aquatic habitats, and some others started to take flight. Not only birds, but the pterodactyls, those are not birds and those are not dinosaurs. They are their own lineage and they are flying reptiles, okay? Mammals during this period, all of the species of mammals that They were not many, they were tiny and usually nocturnal. To what? To avoid predation and competition with the reptiles. And by the end of this period is where we have the appearance of flowering plants. And then we will talk about how important the appearance of flowering plants and insects taking over the terrestrial environment was very helpful for the increases in diversification of these two groups of organisms. Now let's move into the era where we live now, the Cenozoic era that started with the meteorites 66 million years ago. That of course made most dinosaurs extinct. The only survivor relatives of dinosaurs, of T-Rex and all the theropods, are the birds. And it opened the opportunity for mammals to start diversifying more and to start dominating the planet. Now, during this period, climate started to cool. And then about 2.5 million years ago is when we had the last set of glacial cycles during the Pleistocene. All the way to about 15,000, 10,000 years ago, we are in between glacial cycles. So we are living in a warm period of the history of the planet. So during this period, the climate started to cool, but then there was these cycles of cooling and warming, okay? And the primate ancestors of humans started to appear about 6-7 million years ago. We will go into more details about that in a couple lectures. Now, body plans. In body plans, basically, it's just how the animal is divided in terms of their symmetry. When we talk about symmetry, we talk about two main types of symmetry: bilateral symmetry and radial symmetry. And we will see some pictures in a second. When we are talking about radial symmetry, we are talking about dividing an animal in a central axis plane, that whenever you divide that animal, you are supposed to get equal parts of the animal. Usually radial symmetry is present in marine organisms that have sessile adult stages. Okay, so that is commonly the type of symmetry that they are going to present, also like sponges. Bilateral symmetry is basically the one that we observe in most of the animals that we know on this planet, including us. And the bilateral symmetry can be in two forms. If we are standing like this, it's going to be from head to back, and then the other portion is from the dorsal to the ventral portion, like in this arthropod over here, okay? So those are the two main types of symmetries. Ten more seconds. Okay. Questions? No? Let's move into other generalities. Sensory organs is when we start to really see them in full development in animals, and usually they are going to be localized in one portion of the body, usually the anterior portion, like in our case. But not all animals have such well-developed nervous systems. We will see, we will talk about that. Now, tissues. Tissues is going to depend on what animal group we are talking about. Not all animals have complex and different types of tissues like the ones that we have. We will talk about those transitions as we move through this chapter and in particular the next chapter. Some animal groups like sponges are considered to have no real tissues, although they have some epidermis, and we will talk about that later on. But the most general thing that you can mention about tissues is ectoderm and endoderm. So basically the outer portion of the animal and then the inner portion of the animal, okay? But that is like the most general thing that you can say about tissues. When we advance in evolution into more complex organisms, then we have other terms. We have what we call diploblastic and triploblastic animals. So that means either 2 types of tissues or 3 types of tissues. We are animals that have 3 types of tissues. We will talk about diploblastic animals in the next chapter. And also, don't worry about the mesoderm right now. It will become clear once we talk in a few slides. Body cavities, as you know, many animals have cavities, but those cavities are going to be different depending on the number of tissues and the type of tissues that they are going to have. But usually body cavities are going to be filled with fluid that is going to provide properties to the organism to support internal organs, or also to provide support against external pressure so they don't get squeezed, okay? The internal cavities, the technical term that you are going to see is cell, just as it is written up there. So that's the internal cavity. Now, when we talk about a cellum, we are talking about an organism that has 3 types of tissue that are listed there and color coded. Okay, so we have the ectoderm in blue, we have the mesoderm in red, and then we have the endoderm in yellow. Now, when we talk about a cellum, those are cavities that are in between the mesoderm, only the mesoderm, because in this type of animals, the mesoderm, the red color, is going to provide tissue to support internal organs, and that is an example. We are an example of this type of cavity, a cellum, because the muscles, which are part of the mesoderm, they provide certain support to the internal organs. Instead of having all our organs just floating around in a cavity without any protection or type of support, that is the case for other organisms that we call hemocell. We will talk about that in a second, okay? So again, when you talk about the cellum, the mesoderm provides support for internal organs. Hemocell, we also have 3 types of tissue, but now the endoderm, which usually develops the internal organs of organisms, is just floating around. It's like a balloon, and then inside the balloon you put some water, and within the water there are the organs floating around, okay? So, and that space is known as the hemocell, and that is the most common type of cavity in invertebrates. For example, insects, okay? So you can see again that the endoderm doesn't have any type of support by mesodermic tissue. Okay, we will talk about all the groups that have this type of cavity. Now, some of the triploblastic animals, meaning three types of tissue, are not going to have any cavity at all. They are going to be completely flat, and as the name implies, many of them are going to belong to a group of invertebrates known as flatworms or platyhelminths. We will also mention those. And many of the things that are going to be characteristics of this type of animals is that they don't have a full digestive system. They are just going to have a blind side, and many of the metabolic processes are going through to occur through diffusion with the external portion of the body. Now, when we talk about development, we have two main groups, deuterostomes and protostomes, okay? And they have certain differences. All of them undergo development, as we saw in the video last class, but the differences between protostomes and deuterostomes are three main ones. The pattern of cleavage, or how the cells in the embryo are started to be produced when they start to multiply. So in protostomes, we have a spiral and determinate development of cells. And the opposite is going to be in deuterostomes. We have radial and indeterminate development. What does this mean? Determinate means that the cells, once they are produced, they know what type of cells they are going to be. Meaning they are going to be skin cells or liver cells or whatever type of internal organ they belong to. Whereas in deuterostomes, and that includes us as animals, we have indeterminate cells at this 8-cell stage of development, meaning they are like stem cells. They have all the potential to become many different types of cells. The other difference between deuterostomes and protostomes is how the internal structure, the cavity, the cellum is formed. You can see here that the archenteron, which is the technical term for the cavity that it forms in the gastrula stage, here is the blastopore. You can see that the mesoderm forms in an independent fashion from the endoderm, the yellow color here. Whereas in the deuterostomes, the mesoderm is part of the endoderm. And that gives you a hint of why the deuterostomes are going to have what type of cavity? If we belong to the deuterostomes, what type of cavity we are going to have? Okay, cellum or hemocell. Well, maybe you said it right, maybe I didn't, but I call it cellum and or hemocell. Okay, so cellum is the one that we have that has support for internal organs, and hemocell is the one that doesn't have support for internal organs. Okay, and that's mostly for invertebrate organisms. Now, the other difference is what the blastopore during development ends up being. In protostomes, it becomes the mouth, and in deuterostomes, it becomes the anus. Okay, so those are three main differences in embryonic development between these two large groups of animals. Okay, are there any questions? Ten more seconds. Okay. more seconds. Questions? Yes.
I wonder, it just shows the lecture, doesn't show questions.
Oh, sorry, let's repeat that one. That was my mistake. I did too much clicking. Okay, so let's try again. Okay. All right. Questions? No? Okay, so we said earlier that from about 500 million years ago, all the phyla of animals that we have on the planet were already determined, okay? So anybody living today on this planet can date an ancestor back to what period? 500 million years ago? What is the name of that period? Cambrian, okay? Remember, the Cambrian is rapid diversification. And we know all that because we have many types of evidence. In particular is genomic evidence, especially during the last couple decades. But of course, the first evidence that we started to use was morphological. Then, with more advances in technology, better evidence, molecular evidence became available. Now, ribosomal, I'm going to make a pause in here because it's important that you know this. From all this molecular evidence, ribosomal or rRNA genes, those evolve slowly. Anybody knows what are those genes coding for? What do they produce? The ribosomal RNA. Yes, proteins, but those proteins form what? Where does the synthesis of proteins occur? Yeah, in ribosomes, okay? So ribosomes are like factories, okay? So ribosomes are the cell factories producing proteins. Now, if you want those factories to keep working in an efficient way, you need them to be very well preserved in the sense that you don't want many changes to occur in those factories, especially at the same time. So the pace of evolution of genes coding for ribosomes is very slow. So if it is very slow, are those genes most useful to understand the history of life on the planet that is most recent to the present or further back to the past? How would you use molecular markers that evolve slowly?
Those that are further back in the past.
Yeah, further back into the past. So one thing that you have to keep in mind is that not all molecular data is the same, and it doesn't have the same use in order for us to understand the history of life on the planet. If something changes slowly, it's very good to go deeper back into history because it's not changing that much. And ribosomal DNA or ribosomal genes are the ones that have been used in order to identify the large free domains of life on the planet, along with other data, but mostly using those type of slowly evolving genes. If you would like to see changes occurring at the population levels in different populations of the same species on different locations around the world, if you want to see if some populations are accumulating genetic differences, you wouldn't use this gene, because this gene would give you exactly the same. It would be like a clone across the whole set of populations of that single species. Instead, what you use are molecular markers that change faster, meaning they accumulate mutations in a faster way. And there are different molecular markers that you can use to do that. But again, different molecular tools for different questions in relation to the time of the history of life on the planet, okay? So important points, all animals share a common ancestor. Anybody remembers when that ancestor arose, more or less? A ballpark? It was in the Cambrian? Yeah, yeah, over 700 millions. Is that the Cambrian? Neoproterozoic or Precambrian is also commonly known as Precambrian, because it's a huge chunk of time before the Cambrian. Now, sponges are the sister group to all animals, and they are considered to having not true tissue. Okay? According to what we've been discussing, what could give you the idea of true tissue? In order to jump from unicellular eukaryotes to multicellular eukaryotes, what do we need to happen?
I was thinking without the organ system.
Of what?
Organ system.
Yeah, that is part of the process, but if we think about the cells that form a multicellular organism, what do they have to do in order for us to consider them a true multicellular organism?
The cells can't live outside the true colony.
Yeah.
They survive outside of that.
Yeah, basically there is mutual dependency, right? Because one thing is that many unicellular eukaryotes come together for convenience and form a colony, but they can live on their own. In a true multicellular organism, we don't have that. And in multicellular organisms is when we start to see the development of a specialization in terms of different types of tissues. Now again, sponges are considered to have not true tissues, even though they are multicellular eukaryotes, okay? But we will talk more about that. One technical term that you might want to remember is that all animals except the sponges and a few other groups that we will discuss are known as eumetazoans, or true animals. Most of the animals that we will be discussing belong to the bilateral clade, meaning they have bilateral symmetry. And most bilaterians are invertebrates. Okay, so most of the animals that we know of are invertebrates. And the vertebrates belong to the phylum Chordata, but not all within the phylum Chordata are going to have bones or are going to be vertebrates. There are some couple invertebrate groups. Here is a very broad summary of animals. So Metazoa, about 770 million years ago, Precambrian. The basal clade are the sponges, the porifera. Then we have the Metazoa, okay, or animals with true tissues, and we have many different groups, mostly invertebrates. And then when we reach the bilateral, 670 million years ago, this is the period of the Ediacara biota, we start to see three main clades, and we will talk about all of those. The Deuterostomia, that includes us, all the chordates in here, and then we have two larger groups, the Lophotrochozoans and the Ecdyxozoans. We will cover all those groups. So, as I just mentioned, Deuterostomia might include vertebrates and invertebrates. Most of the animals in Deuterostomia are going to be invertebrates. Ecdyxozoans and Lophotrochozoans are all invertebrates. We will talk about the main things. Ecdyxozoans, basically, they are called like that because they have to shed an exoskeleton in order to grow. So the process of changing the exoskeleton, just like spiders, like insects, in order to grow is called ecdysis, okay? And the Lophotrochozoans clade have two main characteristics, but those two main characteristics are not present in every single organism of this clade. One is called the lophophore, which is this kind of ciliated structure that they use in order to capture particles in the water that is passing by, which is going to become their food. And then many groups, like mollusks, are going to develop this type of larvae that is known as the trochophore. Okay? So those are two unique characteristics of the Lophotrochozoans, but not all Lophotrochozoans have them. But let's say that you describe a new organism and you see that during development it presents a lophophore or a trochophore larvae. Then for sure it belongs to this clade. But if you don't see it, then you have to dig more information in order to determine what type of invertebrate organisms you are dealing with. Okay? Ten more seconds. Okay. Okay, when we talk about animal systematics, two questions that are still open and that research scientists are engaged with is to define if ctenophores, you will see some pictures of those things. Ctenophores are the basal or the basal clade of metazoans, and if some group of flatworms known as acoelomates are bilaterals or not. Okay? Broad, simple questions, but we still don't have an answer for them. Now, remember, just this is like a summary for you. Last class we finished the fungi, and the fungi and all other animals shared an ancestor about 1 million years ago. Okay? When we reach the animals, we are talking about over 700 million years ago. Now, if we go a little bit deeper into the animals, what we are going to see at the base is the porifera, which is basically the sponges, and then we have the development of tissue. And at the base currently we believe is the ctenophores. And starting from the ctenophores is what we call eumetazoa, animals with true tissues. Okay? And then further down in time, we arrive to the bilaterals, which is most of the animals, okay? And in bilateral symmetry is where we see the development of the three tissue types, okay? Before this split between the cnidarians and all these other bilaterians, we only have two types of tissue, or diploblastic, okay? Diploblastic organisms, and then after here is the tripoblastic organisms, which are bilaterians. Another small summary. So before the Cambrian explosion, around this time, over 500 billion years ago, we have all the Neoproterozoa going from a billion years ago until the end in the Ediacaran biota, starting in the Cambrian explosion. Then we have the Paleozoic that lasted over 250, 280 million years, and then another mass extinction in here. The largest mass extinction started the Mesozoic period, the dominance by the dinosaurs. Then the other mass extinction in here, 66, 65 million years ago, started the era of mammoths, okay? The most recent, the Cenozoic. And anybody remembers how many extinctions did we have during the Paleozoic, mass extinctions? Because the famous one is the Permian, right? It started the dinosaur era. But before that, we had other extinctions. Anybody remembers how many we had during the Paleozoic, in addition to the Permian extinction? Two, okay? Two more. And one was due to reduce sea levels and also the cooling of the environment. A little bit of glacial cycles, of course. And so most of those mass extinctions occur in marine organisms during the Paleozoic era, okay? Questions? Okay, 10 more seconds. More seconds. All right. Okay. Before we move into the next chapter, I'm going to give you a few minutes to stretch if you need to, or also to go to the restroom if you need to.
like standard. I mean, I just don't.
Yeah, like, I didn't have to finish it now.
They just do it.
I just wanted to let you know, I saw that I don't like your boss. Like they, yeah, yes.
Right. It was just funny because I got a visa. She didn't know what she was doing. And she just punctured the 3 times. And I was like, I don't think that's sanitary. But she's like, it was, I didn't feel it anyway. I was like, I don't think that's sanitary. And then she had a helper. She was looking, she was like, it's okay, it's like tube's faulty. Tubes. It's just funny.
All right, guys, let's resume the lecture. So now we're moving into the chapter on invertebrates. It's a very big chapter. I will try to move more efficiently, just giving you the most relevant information. If you want to dig more details into some groups, just use the textbook. Okay, again, let's start with remembering the tree or the phylogenetic tree of animals. So common ancestor about 770 million years ago. The basal clade is the sponges. Then we jump into the true animals or the Eumetazoa, where we start to see the development of what is called true tissue. And then within the next clade, which is the Bilateria, is where we start to see 3 different types of tissue. Okay? Now, invertebrates is most of the animals on the planet. 95% of animals that we know on the planet are going to be invertebrates, and they are going to be located everywhere where you look on the planet. Very diverse in terms of morphology, so that is not a surprise either. And some of them are going to be microscopic and some of them are going to be huge. We are talking about many meters in length, and I will give you some details on some of the largest that are also parasitic that you can find in the animal kingdom. We have described over 1 million species of animals. 1.3 it says in your textbook, but there are many more described now. There is a new edition of the textbook coming out soon, so that figure is going to increase a little bit, but it is estimated between 10 and 20 million animal species are somewhere around the planet, so many of them probably we will never know them. Now, if we breeze very quickly through the different groups, first we have the porifera of the sponges. We have over 5,000 species, and they are going to be mostly sessile and filter feeders, and they lack true tissue. That is the group of animals, basal one, that doesn't have what we or what physiology is called true tissue. Then we have the Nidarian, 10,000 species, and here is where we have corals, jellies, hydras, but we will go deeper into this part. They are going to have radial symmetry, and they are going to have this gastrovascular cavity. Gastrovascular because they don't really have a true vascular system. It's just going to be a set of a network of vessels that carry water. We will see those details. Then we have Acella, 400 species. They are mostly flatworms. So nervous system is very simple. The digestive system is also very simple, basically like a blind sac. Then we have a group that has only one species that is really weird. You wouldn't think it's an animal, but this thing is an animal. It's called the group is called Placozoan, and it has basically 2 layers of tissue, and this is an oddity within the animals. It doesn't have sexual reproduction. It's basically by budding or by division, a sexual reproduction in this organism. And I am not sure, but I think, depending on the environmental conditions, they might undergo some type of sexual exchange of genetic material, but I'm not completely positive about this group. Now, Ctenophora, or the jellycombs, about 100 species. They have these 8 rows of combs that are the colorful ones that you see, and they are ciliated, and they are the ones that they use to move around in the ocean. Now, those are the basal groups. When we talk about the Lophotrochozoans, one of the big clades of animals, first of all of them are invertebrates. And all these are the groups of Lophotrochozoans. Some of them are very weird, as you will learn. So let's start moving in here. Flatworms or platyhelminths. This is a very species-rich clade, not the one with the largest number of species, but is very spacious, 20,000 species. So they have bilateral symmetry, and as you know from the previous chapter, they don't have a body cavity. They are completely flat. And because of their flatness, their organs are very simple, and many other things are very reduced. The nervous system is simple, and the excretory system is also simple, but we will talk more about those details. Here we have many parasitic species. We will mention more about that. Then we have the other groups, Syndermata. Close to 3,000 species have been described. 2 main groups, one free living that is called the rotifers, and a rotifer is just like a picture in there, and they are tiny. You can see the scale there is 100 micrometers, but they are animals, and they have true tissues, and they have a full set of organs inside their tiny body. And the other group is the Acanthocephalans, which is basically all parasites. Modified, their lifestyle is modified to be just parasites. And they are also known as horny-headed worms. You will see why later on. Ectoprocta, 4,500 species. They form colonies. They form these solid structures, an exoskeleton of carbon. And then they have the classical feature of Lophotrochozoans. They this ciliated structure that is used to capture food particles floating around. Brachiopods, 355 species. They look like many other shells, especially within the mollusks, but they are their unique group of invertebrates of Lophotrochozoans. You can see here that they have the lophophore inside the 2 shells. This is not present in other shell organisms like clams or oysters, okay? So this is unique to the Brachiopoda, okay? Gastrotrichia, 800 species. These are worms, aquatic, that live in the bottoms of either fresh water or salty waters. and they have these bellies covered with cilia that they use for movement, which gives them their basically is where the name derives for gastrotrix, because gastrotrix is the name given to those type of cilia in these organisms. Now we have a weird one here, Cyclophora, just one species, just like the placoderm that we mentioned before, but this one is tiny as well. It lives in lobsters, it's a commensal of lobsters, and it has this weird sexual reproduction system where the male comes into a mature female and it deposits the sperm, but the sperm doesn't fertilize the adult female, but rather the females that are developing within the adult female. Okay? So there are oddities in nature, especially in this type of invertebrate organisms. Well, that's our point of view, right? So maybe for them it's just natural. Nemertia, or ribbon worms. About 1,000 species, and the coelom is also reduced. They have a coelom, but they are mostly flat, and they are all aquatic, and they are predators. And so the only organ that they have reduced is the excretory organs, because they do it more through diffusion. Then we have the worms, like the earthworms, the annelids, about 16,000 species. They are going to be segmented. We will talk more about segmentation when we talk about the arthropods, but the earthworms that you find in your garden belongs to the annelids. Okay, and of course it's not only terrestrial, actually terrestrial annelids are the less number of species. Most annelids are going to have some form of aquatic habits. Then we have the mollusca. Mollusks is the most specious group. It has over 100,000 species, and there are different groups in here, but obviously snails, the clams, squids, and octopuses are the main ones. Okay, so that's a breeze through the Elophotrochozoans, now the Ecdysozoans. Now, in the Ecdysozoans is the group where we find most of the animals. So now I want you to notice something. When we say animals, 95% of animals are invertebrates. So most animals on the planet are invertebrates, okay? But within invertebrates, we have two groups. We have the Elophotrochozoans that we just reviewed, and then the Ecdysozoans that we are about to review. All those are invertebrates, but within invertebrates, the Ecdysozoans is the ones that contain the most number of species, or the richest group on the planet, okay? Now, anybody remembers what is one of the crucial features to say that an organism is an Ecdysozoan? If we have arthropods in there, how do arthropods grow? Exoskeletons. What do you have to do with the exoskeleton in order to get bigger? To shed it, right? So that process of growing by shedding the exoskeleton is ecdysis. And that Greek word ecdysis, that is what it means, and that is what it gives the name to this large clade, okay? So, one group, few species, marine, tiny, the Loricifera, 10 species, and the feature of these invertebrates is that they have these solid plates that are covering the softer body, and the proboscis that you see in here is basically the head that comes out of that armor in order to feed from the water column, okay? Then we have something that looks like worms, but they are not worms like in the case of the flatworms. This is the Priapula, 16 species. They are all marine, they live in the sediment, and they are predators. So they hide in the sediment waiting for the prey to come by. And what is interesting about this group that now in the current time is rare, only 16 species, is that this group was very diverse in the Cambrian period. And there were some of them that were huge in size. We are talking about some meters, and they were predators. The next one is velvet worms, the Onychophora, about over 100 species. They are mostly attracted to humidity. Many species are marine, but so many of them now live in humid environments like rainforests. And they are basically moving through pseudopods, and they have this type of antenna similar like to snails and to what is the ones that don't have a shell that are like snails? Slugs. Okay? Tardigrada. These are becoming more and more famous, about 800 species. They are called water bears. They are tiny. Have any of you heard of these guys? Yeah? What do you remember about those guys? Basically, right? So these guys, they are tiny and they can withstand conditions that are extreme even for the extremophiles, okay, that live in those conditions. They can withstand those type of conditions. Here is just one example, minus 200 Celsius degrees. That is close to absolute zero, and they can survive that. The other reason they have become a full research agenda is because there are experiments where they are shipped out into the space with no protection, like the astronauts. So they receive full radiation, and they undergo this type of hibernating process where they go to sleep, and then when they return to Earth and they are put into nice conditions, they come back to life, and when they analyze their genomes, nothing happened. So they are doing just like nothing happened. So you can imagine that's very important for humans trying to colonize other planets, but we are wimps compared to them because a little bit of radiation and we're already dead. So we are trying to figure out the tricks of these guys in order for us to move around the stars. Okay, at least if we want to move as humans as we look now. There is this guy from Corvallis Medical Center. He has a book called The Next 500 Years, and he does a lot of medical space research. And he suggests that we humans, yeah, we're going to colonize some other planets and probably other stars, but not as fleshy humans, but rather more like bionic or a version of cybernetic humans. That's what he thinks, because of all the challenges that we have to overcome to go to outer space. All right, let's go back to diversity here on the planet. Nematodes is very rich in species, 25,000 species. These are roundworms. Most of them are going to be free living and very important for environment, for a healthy environment. But many species also are parasites, and parasites not only of animals but also of plants, meaning that parasitism has evolved independently in this clade of roundworms many times, okay? And then the king of all animals, the arthropods. We have described more than 1 million species, and of course the estimates are a lot more than that. They are characterized because they have segmented bodies. We will talk about all of those things. So the biggest diversity of animals on the planet is invertebrates, and within invertebrates is in the dixozoans, and within the dixozoans is the arthropods. Okay? Now let's move into Deuterostomia, the other large group of animals. In here we find invertebrates and then vertebrates. We belong to the Deuterostomia, and we have in here just 3 groups. The Hemichordata, about 85 species, which are basically chordates, meaning they have some developmental features that are more similar to us humans than to, for example, annelids or earthworms, even though they look very different. They are mostly marines and they can grow very big in length, up to 2 meters, and they are also type of predatory species. Then we arrive to the chordates. The chordates includes, again, all the vertebrates, but also invertebrate groups. And here we have about 60,000 species, and the two groups that are invertebrates is the lancelets and the tunicates. And that is an adult tunicate that is more closely related to us than to sponges or to other invertebrates. And you will see why. It looks very different as an adult, but as an embryo and during development, there are very specific features that then make it look like a real chordate animal. Then we have the echinoderms or echinodermata, 7,000 species. Here is where we find all the sea stars and dollars and all the sea orchids, echinoderms in general, okay? And they are going to have one feature of them is that they have a hydrostatic skeleton, quote unquote a skeleton, because they don't have bones, okay? And it's called hydrostatic because it uses the pressure of water in order to maintain shape and for movement. All right, let's go into a little bit more details about some of these groups. So we have the sponges again, porifera. These are the basal ones, and they can go from very tiny to very large sponges, several meters. Filter feeders, and they are going to have a cavity, and that internal cavity is where the water coming out from the ocean is flowing inside that cavity, and the cavity is known as the spongiocell. Okay, so that central cavity, the spongiocell, is going to allow certain cells that we will see in a second to capture the particles in that are going to become the food for these sponges. I already mentioned that. So let's go into the important details here. The cells of the cells forming the quote-unquote tissue of the sponges are known as the choanocytes, okay? And remember from the previous chapter, we say that the choanocytes basically look almost equal to the choanoflagellates. And choanoflagellates are single-cell eukaryotes that can form colonies, but they are considered the sister to all other animals. Okay, they are still not considered animals, even though sponges have cells, or it's composed of cells that are almost identical to the choanoflagellates. Okay, so here is the basic structure of the sponge. So we have the epidermis, we have the pores all around the body, all around the epidermis that are going to allow the flow of the water into the internal chamber that then is going to be pushed forward outside of the organism through the osculum. This opening is known as the osculum. Now, if we jump into the choanocytes, what we are going to see is that the choanocytes, this single cell, is going to have this color that is going to capture particles, is going to be absorbed by phagocytosis into the main portion of the cell, and then those phagocytic vesicles are going to be passed into the basal portion of this cell that is the amoebocyte. And in the amoebocyte is where all the metabolism is going to occur. Now there is something that it can be considered like the tissue or the adhesive joining all the different cells of the sponge that is known as the mesohyl, okay? But this mesohyl is more like a glue for giving this form to the organism. It's not like real tissue like the one that we have, that everything allows certain type of communication between different types of tissues, okay? In terms of reproduction, most are hermaphrodites, okay? But in order to avoid self-fertilization, they do something that is known as sequential hermaphroditism, meaning that first they are males, then females, and every individual, independent in the colony where they are living, do it at a different time. So in that way, they avoid self-fertilization, which is good for genetic diversity. Now, during the larvae stage, they develop this larvae that is free swimming, okay? And that larvae eventually is going to sit on the substrate and develop into this sessile adult. Questions? Ten more seconds. Okay. All right, let's move into the cnidarians, which is the next basal group after the sponges or the porifera. Here is where we start to see true tissue, okay? And the oldest, this is the oldest phylum in the animal groups that have true tissue. Anybody remembers how do we call those animals that have true tissue? Is there? Eumetazons, okay? So here is where we have the jellies, the hydras, the corals, all these ones, and they are going to have the gastrovascular cavity that you will see in a single, in the next slide. So they have a single opening that acts both as a mouth and as an anus. And they have two main types of body shapes. They can be either polyps, like in many corals, or they can be the medusa form, which can be the reproductive form of many of these organisms. Of course, the shapes are going to be very different. Now, many polyps in the adult stage are going to be predatory. They are going to have the tentacles, just as in medusa, and those tentacles are going to have specialized cells to capture and process prey. We will see those details in a second. But here is a small video for you to see a polyp-shaped invertebrate capturing a prey.
Commonly called a water flea on the left, and a hydra, a sessile animal in the phylum Cnidaria, on the right. Like most cnidarians, a hydra possesses a ring of tentacles that surrounds its mouth. The mouth leads to a gastrovascular cavity with only one opening. Hydra, like most cnidarians, have highly specialized stinging cells called nematocysts. Some of these stinging cells inject a type of venom that paralyzes the hydra's prey, allowing its tentacles to surround and draw the prey into the mouth. The video shows the daphnia being immobilized and the hydra ingesting the prey into its gastrovascular cavity. The food is then broken down into small particles that are engulfed by phagocytosis by the cells lining the gastrovascular cavity. Present courtesy of Graham Art.
Okay, and here you will see the asexual reproductive process of a hydra, meaning it's just like budding. That is the main reproductive process in this group. Here. You will start to see how a small hydra starts developing from the main stem, and then it's ready to be released. You see, here you can see like the mini-me of the adult one. Okay. So, the other video mentioned the specialized type of cells that these organisms have in order to paralyze, inject venom, and paralyze the prey. Well, those are known as the cnidocytes. if you take one tentacle of these organisms and zoom in, what you will see is here the cell, the nidocyte, right here inside the tissue, and inside the tissue we have what we call the nematocyst, which is an organelle specialized to trigger a harpoon-like structure. So this thing that you see here, like a needle, is actually the trigger. So when the tentacle touches a potential prey or any other organism, so the needle triggers the harpoon, and the harpoon is launched, and it goes inside the other organism. It releases this thread and it injects the venom. So if any of you have had any close encounter with medusa while in the ocean, you know it's painful. So it's the venom is not designed for animals like us, but for smaller animals, but it's really painful. Now the other thing about the gastroventricular portion where they process the food is that it's like a hydrostatic skeleton. It allows the animal to basically maintain the shape, okay? So that's the other function, because they are invertebrates and they live in water. Remember, water has more pressure than air, so they need a way to maintain their shape and avoid being squeezed by the pressure of their environment. And we have two main groups, okay? They are separated into the famous medusoans and then the anthrozoans, which are more the ones that develop into polyps, which include corals and sea animals. I'm going to jump this video because you know these ones. When we go to medusoans, we have three main groups: scyphozoans, cubozoans, and hydrozoans. The first two are like the ones that you commonly see in aquariums. It's just the jellies. And most of these groups of hydrozoans are going to be jumping between the two different forms, morphological forms, polyps and medusa. We will see in the next slide a life cycle, so you can see the different stages. Here, the classical type of life cycle that we started to see with plants. We have the diploid portion and the haploid portion. The diploid portion here is the dominant one. And depending on the group of medusoans, the polyp is going to dominate the life cycle stage, like in here. And we have two types of polyps: the one that has these extensions to capture food, and the other polyp that is the reproductive polyp. So these reproductive polyps are going to produce medusas that are going to be male and female, are going to produce either eggs or sperm, and then during this portion is where they undergo fertilization. The zygote develops and the larvae is going to be free floating on the ocean, and then is going to eventually reach some substrate where it's going to get attached to the substrate and then develop into an adult polyp. But again, how dominant the polyp and medusa stages are depends on the species and the group of medusoans that we are talking about, okay? In the case of the of these ones, the hydrozoans, you will always see this exchange between the two morphological types. Anthozoans occur only as polyps, and they are going to form this hard exoskeleton, which is calcium carbonate, and many of those anthozoans are basically the coral forming organisms or animals that form the corals, okay? Now, this is important. Why? Because the corals, which belong to the anthozoans, are the ones that produce these underwater rainforests. And the underwater rainforests are responsible for a huge diversity of the marine realm. So most of the marine animals that we know of have been described in these underwater forests. But now we have a problem, right? Because many of these forests, underwater forests, are bleaching out and are dying off. Now, corals usually form endosymbiotic relationships with some type of photosynthetic organism that we mentioned in a few lectures ago. Anybody remembers who is the endosymbiont of corals? It's an algae, but it's not the green or red algae. It's another group of photosynthetic algae from the larger group of algae. Diatoms. Okay? No diatoms. Sorry. Diatoms are important. It's other group. Dinoflagellates. Yeah. But diatoms also do photosynthesis, but they have these silica shells that accumulate when they die into the ocean. So the dinoflagellates are the symbionts of the corals, and they are responsible for doing the photosynthesis and provide food to the corals, and the corals provide protection to this endosymbiont. But what happens is that when temperature starts to increase, the dinoflagellates get stressed, and then they leave, and then the coral starts to bleach. After bleaching, the coral is still alive. But if the temperature does not restore, and if they don't get back their symbionts, they eventually are going to die. Okay? So, and of course, you can imagine what happens when a forest dies. Lots and lots of animals go along with that forest. Okay, so that's very bad news, and that's why it's important in terms of biodiversity for the planet. Okay, let's go into a few more details about the Lophotrochozoans. They are bilateral, they are sister to the Ecdysozoans, and I think I already mentioned this, that they have these two features, a lophophore and a Lophotrochozoan larvae, a trophocore larvae, but is not always present. It depends on the group. Now, the main groups are listed in here. We already reviewed more or less how many numbers of species we know of each group. So let's move into the different groups. So flatworms or platyhelminths are going to be mostly aquatic. A few are terrestrial, but mostly the ones that have a terrestrial stage is because they have a developmental stage where they come out of the aquatic environment and into the terrestrial part, but then they always go back to the water. Now, most of them are going to be parasites, and they are going to be very damaging parasites. Some are known as flukes, and the other more famous ones are the tapeworms. We will talk and give some examples for those. Now, one important thing, if you remember, is that they don't have an actual cavity. They have 3 layers of tissue, but no cavity. They are flat. Now, since they are aquatic, flat means better surface ratio of surface to area in their body, meaning they have more surface, more area to absorb things that they need from their environment. And we will see in a second why that is important. Here, what we have is 2 geometrical figures, okay, with the same volume. In this example, 27 cubic centimeters. They have the same volume, but then the surface area is going to be different. The flatter shape has more surface area for the same volume. So many organisms and many organs within complex organisms have evolved the capacity to increase the surface area to have more efficient absorption of nutrients. We have 4 different ways to increase surface area.
One is flattening, which is the one that we are reviewing right now with flatworms. The other one is folding, okay, like the mitochondria in kinetoplastics, the huge kinetoplast of trypanosomes, or here the thylakoid membranes that are responsible for the photosynthetic process in the chloroplast. That increases surface area and makes it more efficient. The other one is branching, like the root system, or projections, heavy projections, like the ones that we have in our small intestine. Okay? Absorption of the nutrients that we consume occur in the small intestine, and we have these projections, like hair-like projections in our small intestine, to be more efficient at absorbing the food or the nutrients that we need for our body. Okay? Questions? No? Now, in terms of excretory systems, flatworms have one specialized network of excretory systems known as the protonephridia. Okay? Some of them are going to have something akin to kidneys, but they are tiny. But the main collecting system of waste material around the body is the protonephridia. It's like a network going through the whole body, collecting all the waste material, and then these protonephridian cells close to the surface in the skin are going to release the waste material. Okay? Two main groups of flatworms is the Catenulida and then the Rhabditophora. And Rhabditophora is the most diverse one, and is the one that is going to contain a lot of free living, but a lot of the parasitic species on these groups. Now, one free living that is become a model organism to understand flatworms is the planaria, shown in here. So you can see that the nervous system is very simple. We have two ganglia in the posterior portion, and then it runs along the body. All along the body, it runs two main nerves all across the body. What is not shown in here is that there are extensions, well, maybe not in planaria, but in many other flatworms, there are like rungs, nervous tissue, like rungs across the whole body, connecting the two main nerves running across the whole body. Also, there are extensions like network-like that connect this main nerve to the surface, to the skin of the body of the organism. Okay, so this is a simplified version. And in the case of planaria, they have this lie detector kind of eyes that they can use across their environment. In the case of this example, they are hermaphrodites, mostly all, let me think, most, not all. There are a few exceptions, and we will see one, but most flatworms are going to be hermaphrodites, and but they are going to avoid self-fertilization, and they promote more outcrossing as well. And there are different strategies that they use. One of the strategies that they use is that the female and the male organs are maturing at different times. Okay, when we are talking about parasites, one famous group of flatworms is the trematodes. And trematodes is the group of parasites that have the most complex life cycles on the planet. They include many host species in their life cycles. They can include up to 5 intermediate host species before reaching the final host. And they are going to be switching between aquatic and terrestrial environments. So they are going, and also they are going to be moving from invertebrate hosts to vertebrate hosts. Okay? So these ones, that are known as flukes or trematodes. One important for humans is the schistosomiasis. Usually that one is a problem still in Africa, and that is due to lack of clean water. Now, this is a simplified, very simplified life cycle of schistosomiasis, and this is one of the exceptions for flatworms because they undergo sexual reproduction. Here you have the male, which is bigger than the female, and the male is going to have this canal across the body where the female is going to lodge. So the female is going to arrive into that canal and then they undergo sexual reproduction. And the technical term of that canal is gynecoporous channel. Now, what happens? Here, the final host is a human, it can be other vertebrate organisms, but what happens is that an infected human is going to release eggs into the environment through the feces. When they release waste material, the eggs of these worms are going in there. They develop this ciliated structure that is known as the miracidium. The miracidium has the cilia to move around the water and then locate the first intermediate host. And for most trematodes, the first intermediate host is going to be an snail. It can be different species of snail, but it's going to be an snail. Once it enters the snail, it will undergo 2 rounds of asexual reproduction that are not shown in here. So 1 round of asexual reproduction is going to produce structures that are known as cercari. Within the radii. Within the radii, another round of asexual reproduction is going to form the cercari. The cercari is the one that is going to be released into the environment from the snail host and is going to be a swimming larvae or stage known as the cercari. And this cercari is going to use chemical cues to find the definitive host, and it's going to be either enter the final host because the final host drinks polluted water or polluted food with this stage, or is going to do active penetration. In the case of schistosomes, they do active penetration. They penetrate the skin. And then they go for several hours into peripheral circulation around the body until they reach the internal organ, where they are going to conduct further development. Okay, this is a simplified life cycle for a trematode. As I mentioned, some trematode species after the snail are going to infect another intermediate vertebrate host like a fish, and then the fish is going to develop a stage known as metacercari, and usually the metacercari of that worm is going to develop in the eye tissue. So basically it's going to reduce the visual capacity of the fish, and that is good news for the worm. That is what the worm wants. Why? Because the final host is a water bird. And what do water birds eat? Fish. What fish is going to be easier to capture? The one that doesn't have good vision, right? Okay, so there are many details about the parasite worm. And this is just one example for humans. A schistosome. And the main worm is Schistosoma mansoni. There are 3 different types infecting humans, and there is one that actually releases the eggs in the urinary tracts of humans. It's the one that produces bleeding from the urinary tracts. The other ones go through the digestive system. Now, other example, tapeworms. So again, they are going to be parasites of vertebrates. In this case, we have the example of humans. Now, they are not going to penetrate or they are going to feed on blood like many others, but they are going to rob nutrients that the host is consuming. So the main body shape is going to be like this. They have a head known as the scolex. The scolex is going to change, the morphological features are going to change depending on the species that we are looking at. And then right behind the scolex, we have the neck. And all the way from the neck to the end of the body, we have different portions known as proglottids. Those proglottids are going to be in different developmental stages. The ones right on the neck are immature, and as you move along the length of the worm, the proglottids further to the back portion of the animal are the ones that are going to be mature. And every single unit, every single proglottid has the capacity to produce a full-grown worm. That's why it's so important to get rid of the whole thing, because just one tiny proglottid that remain inside the organism is going to produce a full-grown adult worm. Now, how they do it? They migrate to the intestines of the vertebrate, in this case a human. They are going to attach in the lining of the small intestine, and then they are just going to be sitting there waiting for food to come by and absorb it. And that is why this type of worms produce a lot of anemia, okay? Because then the host is not receiving the nutrients because the worms are taking the nutrients. Now, they can undergo sexual reproduction, and every one of the proglottids is going to produce hundreds, sometimes thousands of eggs. Okay. Now, there are some types of tapeworms that, depending on the species, are going to be easier to be acquired either to drinking polluted water or to eating undercooked meat. Okay? Are there any questions? No? Okay. So rotifers and acanthocephalans. What we have here is a free living rotifer. So you can see it is tiny. Look at the scale. This scale is 0.1 millimeter. So this thing is just probably a centimeter, less than a centimeter in size. But it has all the structures of an animal, 3 tissues, it has a full digestive system, it has everything to be defined as an animal. And then we already mentioned about the number of species. They are going to be aquatic, and basically what they do in order to acquire their food, because they are sessile, is that they are going to rotate this ciliate, the structure at the top. Let's see this short video so you can see how they do it. So basically they rotate it to create a current close to their mouth, and with that current comes the food particles. Basically I just mentioned all this. What I haven't mentioned about them is that they can, the sexual reproduction main one is not the only one, is parthenogenesis, meaning that the females are going to produce other females without needing fertilization. And that is a type of reproduction that we will see also in more advanced animals, like some reptiles, especially lizards. Lizards, some species of lizards can undergo parthenogenesis. And there is another group of rotifers, the Bedeloidea, which haven't changed basically shape in millions of years. So they have remained like that for millions of years, something akin to what happens with crocodilians and also with turtles. It also happens in these simpler organisms. And one important thing is that these animals can switch to the sexual type of reproduction when conditions are harsh. Why they do that? Because they want to generate more genetic diversity. Remember, when there is more genetic diversity, there is better possibilities for evolution to occur that is going to find an adaptation for the survival of the organism in a population. Now, when I put in here also how they do the genetic mixing of information, in the case of these species that have not changed for millions of years, they also acquire genomic or DNA information from their environment. Basically, when they desiccate, there is some cracks in their skin. And when conditions improve, through those cracks, they can absorb environmental DNA. Anybody remembers what other organism does that, which is a very simple prokaryote organism, or is akin to that? Bacteria, right? How many types of reproduction bacteria has? 3. And what is the name of the one that is similar to this one? Transformation. Okay? Anybody remember the other 2? Yes. Good. All right. The other group in acanthocephalans, the other group of these animals is the acanthocephalans. And in the acanthocephalans, all of them are parasites. Okay? There is no free living organisms in here. And they are also known as spiny-headed worms, and that's because the proboscis that they use to attach to the host is filled of hooks. Okay? So all these hooks are attachment structures that are going to allow the organism to lodge into the host and then start sucking up hemolymph or blood depending on the host that they are infecting. And they, as flatworms, they don't have a full digestive system. It's just a blind sac, and they are not that big. They also have complex life cycles. And many of them, in order to complete their life cycle, they are going to produce behavioral changes into the host. Remember that we have talked before about behavioral manipulation by parasites into some hosts? Well, most of these organisms do behavioral manipulations of some hosts in their life cycles. Okay. Ectopods and brachiopods are the ones that we mentioned that have features that are unique to the Lophotrochozoans. One is, I'm going to put the pictures, it's easier. So this is the ectopods. So they form these solid structures, carbonate features that are sessile, and the lophophore that they use to feed goes in and out of those solid structures for protection. So when they need to feed, they come out, and otherwise they are inside this solid structure. Okay? And one important thing is that they are part of coral reefs. So they are some of the organisms that form these coral structures in the underwater. The brachiopods, as we mentioned, they are shell organisms, but they are not closely related to mollusks, to clams or oysters. And actually the hinge of the two halves has a different direction. In the case of these ones, brachiopods, they open in a dorsal and ventral position. The shell opens like this, okay? It's ventral dorsal, the hinge. And they have one characteristic that no other shell has, the lophophore, which is a true trait of Lophotrochozoans. Now we move into the mollusks, which is a very rich group of invertebrates. Here is where we find snails, slugs, we have squids, we have octopus, and we have also the sea cucumbers. No, that is an echinoderm, sorry, scratch that. So all these ones, many of them are going to develop these calcium carbonate shells. But some of them, especially when we reach more complex mollusks like octopus and squids, they don't have this outer shell. Some of them still have some carbonated type of protection, but it's internal, not like snakes. Okay? Main features of snails are in general mollusks is that they are going to have a very solid muscular foot for movement. They are going to have what we call a visceral mass in the internal cavity. They are going to have a unique structure for them that is known as the mantle, and I'm going to move into the figure so it's easier for you to understand it. So the blue line is the mantle, and that's the one, the living tissue responsible for producing the calcium carbonate shell of these snails. Now we have the visceral mass, which includes all these internal organs. Remember, this is an invertebrate and it has a what type of cavity? Hemocell or coelom? Hemocell. No, but it also has a coelom. Coelom is like the like the cavities that we have. But even though these are invertebrates, they have the two types of cavities. The main one is the hemocell, because basically most organs are just floating around. The only organs that have this protective layer that forms the coelom is the lungs and the gonads. The rest is just floating in the hemocell. And then most snails are going to be aquatic or related to aquatic environments. There are not that many species that are terrestrial. And most of them are going to have this scrapping structure known as the radula that is basically going to allow them to feed on the plant material that they have to scrap from solid surfaces. Most of them are going to have separate sexes, and they have a ciliated, most mollusks, not all, are going to have this ciliated structure that is characteristic of Lophotrochozoans that is known as the chocophore. We already saw some figures for that. Now, when we are talking about clades, we have 8 major clades of mollusks. We have the ones that are known as chitons. Then we have the gastropoda, which includes the snails and slugs. Bivalva, which includes all the shells. Many of you probably eat oysters and clams and all these type of shells. They are mollusks. Remember, they are different to brachiopods, even though brachiopods also have shells. And then we have the famous cephalopoda that includes the squids, octopus, and some others that we are going to see in a second. Now, questions? No? Okay. So chitons. Chitons, they look like that. They have all the main features. They are like well protected. They have all these segments, and they basically scrape algae from the bottom, from the bottom of the oceans or the water bodies where they live. Now in gastropods, we have the snails, we have the slugs, and most of them are going to be marine. Very few are going to be terrestrial. Now, when we look at terrestrial snails, one thing that they are going to not have in comparison to marine species is gills. And if they have gills, gills are fairly reduced, okay? Because they basically don't need them. They do the gas exchange in a different way. But every single gastropod that is marine are going to have gills for gas exchange. Going to jump this part. Bivalves, again, the shells. Now, unlike brachiopods, which are also shells, they don't have a lophophore inside the mouth. They have other structures, okay? So that is one difference. The other difference is that the hinge of their shells, it doesn't open in a dorsal ventral position. It's a lateral position, left-right position. They do like this instead of like this. So the hinge is a little bit different. Some species are going to have some light sensory spots around the body, like in this clamp over here. All these dots over here are like sensory. They are able to sense light. And here is a picture of this mollusk, and you can see that it has a shared trait with snails. It has two types of cavities. It has hemocell, which is the dominant one, okay? And then it has the xylem that protects the heart of the organism, okay? So, again, within mollusks, snails, slugs, and these shells are going to have these two types of cavities, which is not common in invertebrates. And now they are going to use their gill system in order to filter particles that is going to become the food of these organisms. Okay? Finally, we have the cephalopods. Okay, all the famous ones. The one that I haven't mentioned is the Nautilus, which is this one here. And within cephalopods, Nautilus is the only one that has a shell. Okay, the other ones don't have shells. The squids and octopuses, and they are going to have these modifications of the extensions of the body. And many of the extensions of the body are going to be used either to paralyze prey because they can inject with specialized cells, or just to grab the food that they are going to consume. Their mandibles are very, very strong, even though they are not apparent. And other very important thing about cephalopods is that they have very well developed visual system. So their eyes, even though they are analogous to our eyes, they evolved independently. We don't have a common ancestor for the same type of eyes. Their eyes are very acute. Their visual sense is very good, and in the case of the squids, is even better than our visual capacity. Now, in terms of circulatory system, they don't have a full circulatory system. And especially when we reach Nautilus. The Nautilus are more similar to the extinct ammonites, and the ammonites were more related to Nautilus, but they are now extinct, and they went extinct also along with the dinosaurs during the mass extinction of the Cretaceous. Okay, we are going to finish with this lecture with this question, and then we will continue next week.